Welcome back. We are at the end of our discussion on photodynamic therapy or light activated therapy for that matter, and in this course we are going to discuss the future of photodynamic therapy and the new therapy, light based therapy that is coming up, which is called photothermal therapy, PTT. Let us go on with it. So, what are the future? We have thus far seen what photodynamic therapy can do, what PDT can do. I am not going to repeat it once again. I have been repeating it for the past 4 chapters. So, by this time you should know what PDT is and how it works. And so, what exactly we envision as our plan in the future, where PDT can be improved or where PDT can be optimized. So, the present research, well, from a normal point of view, you can understand that PDT is a truly multidisciplinary research, right? It is creating huge amount of opportunity for biomedical scientists, chemists, physicists, engineers and medical practitioners. Engineers needs to know how to shine light at areas which are concealed. Medical practitioners such as oncologists, dermatologists, ophthalmologists are directly beneficiaries of PDT. We need chemists and pharmacists to basically develop drugs that are highly selective, and upon their work being finished, they are getting quickly excreted out of the body, so that you do not have to wait for 4 to 6 seconds. Physicists need to know what exactly is the physics behind all of it, how the different singlet and the triplet states are forming, and how are the triplet states resulting in the unpaired electrons are resulting in creation of radical species, which is causing problem. And if we can create it somehow differently, so instead of shining light, can we have a trigger based mechanism in which a drug is getting attached to a specific cell, an abnormal cell, and say after a time period, just by itself automatically it converts there is an intersystem crossing happening and a triplet state is generated, which creates reactive oxygen species. In phosphorus or in phosphorescence, you do not have to shine external light into it, or do you? Remind me that. We have discussed phosphorescence. Do you think an external light needs to be shined to create phosphorescence? Maybe at the initial stage, but what happens at the late stage? Can you see phosphorescence when the light is switched off? Recall your previous lectures. So, can we have something like that, where you do not need an external source of light all the time? And we need to discuss the PDT's effect on different cellular mechanism, not just simply destruction, but can we manipulate, can we modify, and can we cause some sort of a gene expression. So, remember CRISPR? We discussed that in the genetics, that you are using genetic engineering, this CRISPR based technique in which the gene mutations are removed or manipulated or changed. CRISPR is coming up very strongly, and I think it has already been successful in curing I think sickle cell anemia or hemophilia. Just check it out. These are the genetic diseases. Large section of India's population suffer from sickle cell anemia, hemophilia, where these are genetic diseases where the blood cell cannot retain enough amount of oxygen, or the blood cells have difficulty clotting. So, these are genetic diseases, but the genetic diseases forms because of a particular DNA producing some sort of a protein. The gene expression is happening, and that protein results in creation of mutated red blood cells. Using CRISPR, you can target that particular gene. Using CRISPR, you can target that particular gene and thereby cure it, and CRISPR has done that. Can we do that with PDT as well? Okay. So, you inject some amount of drug, sooner or later that drug get attached to specific genes inside your body, inside your cells, inside your nucleus, and they then destroy the bad genes, something that is causing you, making you prone to, I don't know, cancer or diabetes or hemophilia or any other kind of genetic disease. Can we do that where PDT is applied with genetic engineering? So, you obviously have CRISPR technology, but can we generate a parallel genetic engineering technology using PDT, or can we combine PDT with CRISPR? So, these are some of the debates, some of the discussion that is going on in the current research. I keep on hearing this from students, what are the hot topics, what I should write a paper on a specific project. My institute has asked me to write a specific paper, a specific journal on a hot topic on biophotonics. So, these are your hottest of the hot topics, just fresh out of the oven. It cannot get any hotter than this. Look into it. You are combining genetic engineering with physics, electronics engineering, of course, optoelectronics basically, photonics, and biomedical research with direct application in medicine, oncology, cancer detection, ophthalmology, as well as several genetic diseases that you form. These are some of the latest topics that we are working on. We also need to work on tissue oxygen level limitation. So, remember, when we basically the idea is generation of raw species, right, but the raws keep on destroying the immediate surrounding cells, and after some time you know about this process that the singlet oxygen can go on, can get quenched or bleached, because this phosphorus species or this species, they can get affected by the nearby surrounding area, and the singlet oxygen species or even the triplet oxygen species, how long will they continue? They can get exhausted, leading to shrinkage of its radius. It's actually a problem of dosimetry. I understand what dose, how much amount of tumor can be, can it affect, how much, what size of tumor it can affect. Thereby, at this present moment, we are trying to work on light fluence to slow oxygen consumption, providing PDT treatment in oxygen enriched areas, some areas, say for example, your lung, your bronchial tract, which is already oxygen enriched, and development of oxygen independent photosensitizer, which does not necessarily generate reactive oxygen species. It does not generally develop or generate oxygen species. You need to destroy what is your ultimate aim? You need to destroy the nearby abnormal cells, and yes, generation of raw species will destroy it, but is this the only way to destroy that area? We have therefore developing various new sets of photosensitizers which are operating at near infrared frequency, thereby less harmful. One photosensitizer can take multiple photons, they have highly selective rate, and they have very low retentivity. These are the topics, but what I want to actually discuss is if we do not generate reactive oxygen species, what do we do? Well, there are other new light sources, nano lasers, which are cheap, compact, and real time monitoring. This you can read at your own leisure, but I want to quickly go into the process of photothermal therapy. Photothermal therapy, where you are simply generating heat. You are simply generating heat, and the heat is destroying the nearby cancerous cell. There is no requirement of generating oxygenated species. So, most of the photothermal therapy these days are being done with gold nanoparticles. I have heard or I have read work on two-dimensional materials, especially graphene oxide. Maybe black phosphorus has also been used. If you do not know two-dimensional materials, do not worry. I think in the 11th chapter, where nanoBioPhotonics is coming up, I will discuss two-dimensional materials. Two-dimensional material, just it has length and breadth 1 atom thick, and hence we call it two-dimension. The thickness is very small, 1 atom, large surface area. So it's, but what we basically do in photothermal therapy, we use gold nanoparticles. These gold nanoparticles are very small in size, much smaller than the wavelength of light. These gold nanoparticles get attached to specific areas of the cancer cell. Again, I told you how it can be done. Thiolation, antibody, several mechanisms are available in which a part of gold nanoparticle, a sulfur based part, especially the thiol part, attached with the gold. Another part gets attached, thiol, octadecathiol, or these kinds of chain molecules, they can attach with the biological component. And once, so these are the cancer cell, these are huge nucleus, and these are the cancer cells, and you have infiltrated it with gold nanoparticles. So these are nanoparticles, these are gold nanobodies or nanoparticles, and their size are ultra small as compared to the entire cellular structure. So these are the cancerous cell which has been infiltrated with gold nanoparticles. You are shining light onto gold nanoparticles. The electrons, the plasmons, the conductive electrons inside the gold nanoparticles agitate and they relax, resulting in creation of heat. Basically, non-radiative transition or non-radiative relaxation. They generate phonons or vibrations or heat, as I have been telling you. So this is the mouse who have been injected with gold nanoparticles. They have accumulated inside its cell, and you are then shining light onto it externally. So this is the laser source which is shining light onto it, and unlike PDT, photothermal therapy does not require oxygen to interact. It can use longer wavelength light, which is less energetic and therefore less harmful to other cells and tissues. All it is doing, all the light is doing, is simply exciting the electrons that are available, conducting electrons that are available in the gold particle to get excited. The electrons are jumping at different energy levels, and we can, depending on the nanoparticle size, of the gold particles, we can generate them to we can send them at different energy level. So whenever these electrons are returning back to its original position, they return back by phononic vibration. They do not generate, everything does not fluoresce. Remember, there are non-radiative relaxation as well. These are the gold particles where non-radiative relaxation will happen. Non-radiative relaxation meaning phononic vibration, which in other words can be considered as heat. So basically, you put a metal particle into the cell. The metal particle, upon getting excited by light, generates heat. Light is getting converted, so you are heating up the metal. Simply you are heating up the metal is heating up, and it is dissipating the heat in the surrounding regions. The heat dissipated is strong enough to destroy either completely or partially some part of the bad cell, either mitochondria or cell membrane, thereby resulting in apoptosis or necrosis. Your work is done. It can use longer wavelength of light. It does not require oxygen species to create, because oxygen species can destroy other areas as well. It simply ends an oxygen rich radical. It needs to eat something, so it might attack the other areas. So photothermal therapy is strongly coming up, where you have cut out the middleman, which is the It does not require the oxygen to interact with target cells or tissues. It simply generates heat, a very, very localized heat, a very, very localized heat that destroys certain cellular components. All you need is to target certain areas to destroy. Cell membrane is good enough, mitochondria is good enough, nucleus is good enough, and your work is done. So, this is another hot of the hot topic, literally photothermal. So, light is generating heat. So, hot topic, light based hot topic that is very strongly coming up. I myself am personally invested or would like to be more personally invested in working on photothermal and photodynamic therapy, PTT and PDT, using two-dimensional materials. This is something which I would strongly be looking forward to doing a research in my future. So, that basically brings me to the end of this entire module of light based therapy. I hope some amount of this is clear. Remember, this is an entire field in itself and very strongly merging, and maybe it will become standardized pretty soon, where several cancer treatment, instead of doing chemotherapy, where a drug is being induced to attack the cancerous cell, light will be sent to generate either localized heat or localized oxygen species, and thereby treat the cancer. So, an alternative to chemotherapy, and obviously an alternative to surgery. So, hope some of the topics were clear. Please send me your feedback, please send me your comments, please ask me questions on the forum. I will try to answer them as soon as possible, and I will see you next week for a different module. Thank you very much.